DNA structure and function. Genetic engineering in gene editing. DNA, deoxyribonucleic acid, is a double-stranded nucleic acid that carries the genetic information of all living organisms. DNA is made up of 4 types of nucleotides, each consisting of a sugar molecule, deoxyribose, a phosphate group, and a nitrogenous base. The 4 nitrogenous bases found in DNA are adenine, A, thymine, T, guanine, G, and cytosine, C. The sequence of these bases in DNA encodes the genetic information that is passed down from one generation to the next. The structure of DNA is often described as a double helix. The 2 strands of DNA are held together by hydrogen bonds between the nitrogenous bases. Adenine always pairs with thymine, and guanine always pairs with cytosine. The specific order of the base pairs along the DNA molecule is what determines the genetic code. The function of DNA is to store and transmit genetic information. This information is used by cells to produce proteins, which carry out a wide range of functions within the body. The process of protein synthesis begins with the transcription of DNA into RNA, ribonucleic acid, which then directs the synthesis of proteins. The first step in protein synthesis is transcription, in which a section of DNA is copied into RNA. The enzyme RNA polymerase binds to the DNA molecule and separates the 2 strands, exposing the nucleotide bases. The RNA polymerase then reads the sequence of bases on the template strand of DNA and adds complementary RNA nucleotides to the growing RNA strand. The newly synthesized RNA molecule is a copy of a specific section of DNA and is known as messenger RNA, mRNA. Once the mRNA molecule has been synthesized, it leaves the nucleus of the cell and enters the cytoplasm. The mRNA molecule then binds to a ribosome, which is the site of protein synthesis. The ribosome reads the sequence of codons, 3 base sequences, on the mRNA molecule and directs the synthesis of a specific protein. The process of protein synthesis is also known as translation. Each codon on the mRNA molecule codes for a specific amino acid, which is the building block of proteins. As the ribosome reads the mRNA sequence, it matches each codon with the appropriate amino acid and adds it to the growing protein chain. Once the ribosome reaches a stop codon, the protein synthesis process is complete, and the newly synthesized protein is released. The specific sequence of nucleotides in DNA determines the sequence of amino acids in a protein. Because different proteins have different functions within the body, variations in DNA sequence can lead to changes in protein structure and function. These changes can have profound effects on an organism's traits and characteristics, and can even cause genetic disorders. In addition to its role in protein synthesis, DNA also plays an important role in cell division. Before a cell can divide, it must first replicate its DNA to ensure that each new daughter cell receives a complete copy of the genetic information. The process of DNA replication is a complex and highly regulated process that involves the unwinding of the DNA double helix, the separation of the 2 strands, and the synthesis of new complementary strands. DNA replication begins at specific sites along the DNA molecule known as origins of replication. The enzyme DNA helicase binds to the DNA molecule and unwinds the double helix, separating the 2 strands. The separated strands then serve as templates for the synthesis of new complementary strands by the enzyme DNA polymerase. As the DNA polymerase adds nucleotides to the new strand, it checks for errors and corrects any mistakes. In conclusion, DNA is a double-stranded nucleic acid that carries the genetic information of all living organisms. The structure of DNA is a double helix, with the 2 strands held. Genetic engineering and gene editing. Genetic engineering and gene editing are 2 closely related technologies that allow scientists to modify the DNA of living organisms. These technologies have the potential to revolutionize fields such as medicine, agriculture, and biotechnology, but they also raise important ethical and safety concerns. Genetic engineering refers to the deliberate manipulation of an organism's DNA to introduce new or modified traits. This can be done using a variety of techniques, including the insertion of new genes, the deletion of existing genes, or the modification of gene expression. One of the most common methods of genetic engineering is the use of recombinant DNA technology. This involves isolating a gene of interest from one organism and inserting it into the genome of another organism. The resulting organism, known as a transgenic organism, will express the gene of interest and exhibit the desired trait. Gene editing, on the other hand, is a more precise method of modifying DNA. It involves making targeted changes to the DNA sequence using tools such as CRISPR-Cas9. This technique allows scientists to cut DNA at specific locations and introduce new genetic material or modify existing genes. One of the most promising applications of genetic engineering and gene editing is in the field of medicine. These technologies have the potential to cure genetic diseases, such as cystic fibrosis and sickle cell anemia, by replacing or repairing faulty genes. Gene editing may also be used to develop new treatments for cancer, by targeting and destroying cancerous cells. In agriculture, genetic engineering has already been used to develop crops that are resistant to pests and herbicides, as well as crops that have improved nutritional profiles. These modified crops have the potential to increase yields and reduce the use of harmful pesticides and herbicides. However, genetic engineering and gene editing also raise important ethical and safety concerns. There is the potential for unintended consequences, such as the spread of modified genes into wild populations or the creation of new and unknown diseases. Additionally, there are concerns about the ethical implications of creating genetically modified organisms, as well as the potential for discrimination or inequality based on genetic traits. As such, genetic engineering and gene editing are highly regulated in many countries. In the United States, for example, the USDA and the FDA have strict guidelines for the approval and use of genetically modified organisms in food and agriculture. In addition, there are ongoing debates about the appropriate use and regulation of gene editing technologies. In conclusion, genetic engineering and gene editing are powerful technologies that have the potential to revolutionize fields such as medicine and agriculture. However, they also raise important ethical and safety concerns, and their use must be carefully regulated and monitored to ensure that they are used in a safe and ethical manner.